Hello and welcome to Bioru's video lessons, genetic additions. So today we're going to be talking about genetics, and to start talking about genetics you have to talk about genes. So genes, probably when you hear that word it comes to mind DNA or other kind of nucleic acid with the genetic information. Genes is something related to genetic information. Another way of thinking on genes is also thinking on chromosomes. So this image here is one chromosome in human beings, one of our 46 chromosomes, which is a condensation of DNA, condensation of genetic material, lots and lots of DNA coiled up here. And in here you have genes. For example, here you have the gene 11p15.5, here you have the gene 11p14.2, here you have the 11p11.12, there the 11q 13.1 and so on. You have some genes located here in the middle of this tiny little piece of genetic information, the chromosome, one of our 46 chromosomes. So, but what is this gene? So in that place over there, the 11p15.5 is the HBB, is a gene responsible for making hemoglobin. There we have another one called TYR that you're going to be seeing in this class. So pieces of genes, they are pieces of information. So genes are fragments of information. So let's start with that definition, okay? Here, as I'm showing the chromosomes, I already show you that you have a P and a Q. Those are the arms of chromosomes. When we see chromosomes later, you're going to see that you have a small arm and long arm, the P and the Q. But let's not focus on this right now, okay? So, human chromosomes like this here, chromosome 11, have some loci of genes, some places of genes. You have the HBB in the 11p15.5, you have the TYR in the 11q 14.3, so those are loci, places of genes, okay? So this here is just to give an idea. And so you have the information for HBB, the hemoglobin over there, and the information of TYR in that one over there, okay? So let's talk about genes then. We have here a piece of DNA that's suffering replication. We saw this before when you studied nucleic acids. This piece of DNA is being replicated. You have the original one, which is in green. You have the new one, which is in blue. As you remember, DNA replication is semi-conservative, maintains half the old one and makes half new. And this DNA is being copied over there. So here you have the original and the new copy. They are complementary. If you have an A, you have a T. If you have a T, you have an A C, G, a G and a C, and so on. The original is over there, okay? And in this case, it was copied correctly. But sometimes the DNA is copied incorrectly. You see there in the bottom you have the original, but on the top, instead of putting a pink, it put a yellow. So in this case here, you have a new copy, but this is with a mistake, which you're going to be calling a mutant. So you have a mutant piece of DNA. In this case, the information is being changed. So you're going to be changing the gene. There will be a change in the information contained in that gene. Okay, so there is the place where we had the mistakes. This piece of information that codes for a characteristics, that you can have the original and you can have a mutant one, you have variation, you're going to be calling allele. So in this case here, you have a GATTCGATCTTA, a sequence of letters. There's a part of a gene that is going to be coding for a polypeptide. So this is already bringing us to one important information. One gene is one polypeptide. A gene is a piece of information that codes for a protein or a polypeptide, a piece of a protein. Okay? So in this case, we have a polypeptide, which is now a sequence of amino acids. All of them are great, the original ones. And this polypeptide is a protein that actually, to be more specific, is an enzyme called enzyme A. This enzyme A is the enzyme responsible for converting a certain substrate into a pink pigment. This is a hypothetical example, but here, for example, we have a substrate that was green, and with the use of this enzyme A, this pigment, this substrate the green one, is transformed into a pink pigment. So something is going to become pink. Okay, this here is the regular one that we have. So in the end, we have pink petals, for example, in flowers. So if you have the allele 1, you produce a polypeptide, that is the enzyme A, that produces that makes that reaction to happen, and you're going to end up having pink petals. So one allele is a polypeptide, but also one allele is something responsible for making a certain characteristic. So a gene, so you have a genetic information that in this case codes for a certain polypeptide that's going to be coding for a certain characteristic. In this case, the color of petals in flowers. Okay, so we have there the pink flower. Alternatively, as we saw that we had a mutation before, we can have an allele 2, which is a slightly different version of that gene. So GATTCGTTA. So there's a change there. That C over there was changed by a T over here. So we have a variation in there. So the allele number 2 is a variation of the same gene. It's the same gene, same piece of information that's going to make a polypeptide, but there are variations, and these variations, that's what we're going to be having now. An altered polypeptide. So basically we make a variation of that polypeptide. In this case, an amino acid has been changed. And change this amino acid, instead of producing what we call enzyme A, we produce a variation of the enzyme that I'm going to be calling enzyme B. Actually, enzyme B is enzyme A is slightly different. But because it's slightly different, this enzyme now converts substrate not into pink pigment but into blue pigment. Okay, so it's going to get there the same substrate green and now making it to become blue. So what? So blue is going to be making blue petals, and you have the blue petals over there. So this here is to illustrate to us what is a gene. A gene is a piece of information that codes for a polypeptide. A polypeptide. In this case here, the polypeptide that produces a certain enzyme that catalyzes a chemical reaction involving pigments. So one gene, one polypeptide. But also in genetics, one gene is going to be one characteristic, the color of the petals. So one gene, one polypeptide, one characteristic. So this here is what we're going to be defining as a gene. Okay. To illustrate, we have this gene here called TYR, which is the gene that, when it's not working properly, is responsible for a characteristic called albinism. Okay. Here we have a child that has an albinism with her family. So basically the family is pigmented, they have melanin in the skin, but this kid has very little melanin in the skin, so this we call an albino. How does this work? How albinism work? Albinism is going to be working by this gene, TYR, that when it's working properly produce an enzyme called tyrosinase that is responsible for a metabolic pathway that ends up producing the protein melanin. Melanin is the protein that gives color in the skin and hair and the eye and whatever. So in this case here, this tyrosinase is producing melanin. Okay, so the father of the kid produces melanin, so he's normal. But if you have a TYR that's defective, so it's not working properly, so you produce a defective tyrosinase, the tyrosinase don't work properly, so what's going to be happening? You're not going to be able to produce melanin. And in that case, you're going to have a much lighter pigmented skin because you don't produce the melanin protein that protects us against ultraviolet radiation. So this is one example of a gene. One gene, one protein, and this protein, one characteristic. In this case, producing melanin or not producing melanin. Okay, one gene, one characteristic. Another example that you need to know, especially if you are IB biology, you need to know, is the sickle cell anemia. So sickle cell anemia, we have mutation that happens specifically in the place that produces hemoglobin B, and you have a DNA that you have a GAG that is going to be in the transcription making a GUG, GAG makes CTC, the CTC makes a GAG. So GAG makes GAG back again. And this here, in translation, produce a polypeptide that you have in this codon here, an amino acid glutamate. So GLU. So this here is a normal one, okay? Normal hemoglobin B. So if you have a mutation, and there was a mutation many years ago, we produce what we call a sickle cell anemia mutation. So basically there's a variation there. Instead of GAG, we have a GTG, and that we have a complementary CAC that as a result is going to be making the RNA a GUG. So to remember, GAG in the messenger RNA becomes GUG in the messenger RNA. Okay, so what? So what that in translation, GAG produces GLU, no glutamate, and GUG produces valine. There is a change in one amino acid. So you have a mutant hemoglobin. This hemoglobin is going to be different because instead of a GAG, you have a GUG. Instead of a glutamate, you have a valine. So let's put this here into a big picture for you to see how the normal hemoglobin looks like. You have a series of amino acids over there, and the sickle cell anemia has the same series of amino acids but with one variation. Instead of a glutamate over there, you have a valine. So what's the consequence of that? The normal hemoglobin is a globin, it's a roundish protein. So this roundish protein, works beautifully and nicely. The sickle cell hemoglobin is going to be a little bit different, has a little bit different shape. It still looks like a globin, yes. However, the beta globin, it packs together with another beta globin and two alpha globin, making a bigger protein, a globular protein, while the variant, the beta globin, also makes this kind of variation, but the normal hemoglobin is a globin, a true globin, and makes this roundish protein, and they run free inside the red blood cells. And unfortunately, the sickle cell anemia mutation produces beta globin that link to each other. And when they link to each other, they make a long fiber. As a result, the normal hemoglobin makes red blood cells to have roundish shape, so they can pass through our tiny capillaries without getting clotted, without causing clots and thrombosis and lots of other problems. And they work beautifully. And the sickle cell anemia have fibers of hemoglobin inside. The red blood cell is not rounded anymore, it's going to have a sickle shape, and this is going to be kind of elongated, which don't flow very well in tiny little capillaries, causing blood clots, causing thrombosis and lots of other problems around. So this here is one single DNA mutation that causes a variation in this gene, the gene for the beta globin, that's the gene for the hemoglobin, to be slightly different. You have two alleles, the normal globin and the sickled hemoglobin, and that changes entirely the life of people. Okay, so here just to illustrate to you what is more or less a normal red blood cell, and here a sickle, and here, look, here you have a beautiful shape of a sickle, and this is going to be making the red blood cells not to flow very well in the blood. So here you have, this look pretty okay, pretty good. This looks roundish, but you also have here elongated ones, and this here will make the blood not to flow properly, and then the person to have some health consequences. So, which are the consequences of having sickle cell anemia allele, the variant for this gene? You can have thrombosis, you can have blood clots, okay? You can have circulation issues. Your blood's not going to be circulation very well. And because of that, you may have oxygenation issues. That's why it's called an anemia. It has nothing to do with the lack of iron, which is the common anemia that in this case is for problems in circulation that leads to oxygenation problems as well, okay? It can cause pain, and if it's left untreated, and in the severe cases, it can lead to severe disability and can even lead to death. So it's not something good, okay? So this here is the point. You have there the unrestricted blood flow in the normal hemoglobin, and you have here the blocked one, which causes many health problems. So, how come that this gene, this allele actually of this gene, wasn't wiped out by natural selection? Because if people have this gene, they suffer, they have problems, and they may even die. How come they reproduce? How come they survive to be able to? Natural selection should have eliminated this disease long time ago. And here comes something very interesting, because this disease is something very, very, very bad. However, it can bring some advantages that in some places, like in Africa, it can be something good. This here is the distribution in the in blue there. We are painting where we have happening the distribution of malaria. So these are places that have lots of malaria in the African continent. And these here are the places where you have sickle cell anemia in the African continent. Do they match? Is there an overlap in here? If I put one on top of the other here, look, are they overlapping? Yeah, for most of it overlaps a lot. Not entirely, but for most it overlaps a lot. What happens? If you have sickle cell anemia, especially if you have the less strong, the less problematic sickle cell anemia, we'll learn more about this later, you don't have so much symptoms of sickle cell anemia, because it's the mild variation, but you also don't get malaria. And in the African continent, it might be bad to have sickle cell anemia. If you have the severe form, you're not going to live past your 20s. But if you have the mild form, you can live more or less past your 20s. And very important, you're not going to be dying from malaria that kills millions of people per year in there. What's the alternative for sickle cell anemia? Dying from malaria. If you have sickle cell anemia, you don't develop malaria. The Plasmodium vivax, the protozoa for the malaria, don't manage to develop itself and grow and reproduce inside the defective sickle cell anemia blood cells, red blood cells. So it causes protection. This is natural selection acting today in humans. So this was an overview about genes. We're going to be discussing more about genes in class. I hope you got the idea of what's a gene, one polypeptide, one genetic characteristics. So this here is a gene. I hope you had fun. Do the quiz now to test your learning, and I see you in class. Bye-bye.